welcome back to Biostat Squid. In my previous video, we covered the basics of DGE analysis for single-cell RNAseq datasets. Now it's time to put that knowledge into practice with a hands-on tutorial in R, where we will perform both differential gene expression analysis and data visualization. So if you're ready, let's dive in. In this tutorial, we will walk through how to perform differential gene expression analysis, or DGE, using SORA in R. We will explore three methods: Wilcoxon test, MAST, and DSEQ2. We'll use Wilcoxon test for a quick and easy, very rough first differential gene expression analysis between clusters of a single sample. Then we'll move on to Pseudobulk based differential gene expression analysis using DSEQ2, which is the golden standard as we mentioned in the previous video. And we'll also try the mixed effects model approach using MAST. We'll use a publicly available interferon-stimulated dataset from SORA data. Each method has its strengths, and we'll highlight how and when to use them, including customizable parameters. We'll apply all these methods through the SORA package, but you can also apply these methods directly through their native packages for more control. Nice! So let's begin. We'll start by cleaning the R environment to avoid conflicts from previously loaded objects, then configure R options to improve console output, avoiding scientific notation and truncated tables. Next, we'll load the key libraries, so SORA for single-cell RNA-seq data handling, SORA data to access our sample dataset, then DSEQ2 and MAST for statistical modeling, and tidyverse for data manipulation and plotting. Setting a random seed ensures that results are reproducible, especially for steps like clustering and dimensionality reduction. So we check if the interferon-stimulated dataset, known as IFNB, is installed. If not, SORA data will download it. We're working with peripheral blood mononuclear cells, or PBMCs, from lupus patients, comparing unstimulated, so control, versus stimulated or interferon-stimulated conditions. This dataset is particularly valuable because it includes multiple donors, so it will allow us to address the issue of pseudo-replication properly and do differential gene expression analysis across two conditions. But again, this is only one example. You might be working with two or more time points or different samples or other conditions which you want to compare. You might also have batch effects which you need to take into account. So this is only a standard script for a case scenario. Make sure to adapt it to your own situation. Great, so then let's define an output path where the results like marker genes or visualizations will be saved, the results of our differential gene expression analysis. So I will define it based on my project path and then just add subfolder DGE and then results. Again, adapt this to your own setup and your preferences. So I'll just create that output path. You can also create this folder manually, of course, but I would recommend separating the raw data, your scripts, and your results into different folders. Okay, so let's load the data and have a quick look. It'll take a tiny bit to load. There we go. So there's 14k genes and 14k cells, more or less. I'll just show you the head of it to see what we have. So we have our cell IDs, then the original identity, which will point us to the original kind of data set, so control or stimulated. Then we have the end count and end features, so the number of transcripts and the number of genes per cell. Then we have stim, which tells us exactly if that cell belongs to the stimulated or the control data set. So let's have a look at how many of each kind we have. So there you go, approximately half, so 6.5k control cells and 7.4 stimulated cells. Great. And then we have the Seurat annotations, which have annotations for our cell types. Fantastic. So we'll run the dataset through the standard preprocessing steps. So normalization, variable feature selection using the VST method, then data scaling, principal component analysis, and UMAP for dimensionality reduction and visualization. We will identify cell clusters using nearest neighbor graph-based clustering, and assign our identities based on the Seurat annotations. So you might have a different column called cell type. Great. So you can check my other videos for more details on these steps. And of course, when working with your data, you'll probably have to fine-tune certain parameters. But for the sake of this tutorial, we'll just go with a standard analysis. Great. So we can also run table to figure out how many cell types per condition. So we want them to be more or less even for our comparisons. And then we can use UMAP plot to visualize cells by cluster and by stimulation condition. So we can see the different populations, the different cell populations over here. Let me see. So we have our different cell type populations here. You can see our cells separating by condition. We don't strictly need integrated cells to compare conditions in single cell RNAseq, but integration is often highly beneficial and sometimes necessary depending on your experimental design and your analysis goals. In our case, both conditions were processed in the same batch with identical protocols. The technical effects are minimal between conditions, and the conditions, both control and stimulated conditions, as we saw, are well balanced across batches. There's actually only one batch. So we're only doing simple differential gene expression analysis, so we won't bother with integration. However, if any of these characteristics applies to you, then integration becomes important, and I would recommend integrating both datasets before doing differential gene expression analysis. Okay, great. So now we're ready to start our differential gene expression analysis tutorial, and we'll start with our Wilcoxon test. So the Wilcoxon rank sum test is Seurat's default method for identifying differentially expressed genes between groups. It's non-parametric, making no assumptions about data distribution, and works well for identifying genes with large effect sizes. In our example, we'll compare CD14 and CD16 monocytes within the control group. So we're only subsetting the control group to compare CD14 and CD16 monocytes. Just quickly plot that. So these are our control group cells, and we're comparing the CD14 mono and where are they? CD16 monocytes. So that would be these two groups. So great, the find markers function handles this, and it has customizable parameters like the min percentage of cells expressing a gene, the log to fold threshold to filter out genes with low fold change, and many other parameters. You can read more about this function by running find markers. There you go. So you can check if any of these parameters applies, as in if you want to fine tune any of these parameters. You can, we'll see that later, but you can also modify this test.use to use other statistical tests supported by Seurat, such as bimodal, ROC, or T, depending on your data distribution. But let's run find markers, and we'll visualize the top 10 markers. We'll print them out. So that's a lot of zeros. So it would give you a table essentially with the p-value, the average log 2-fold change, the p-adjusted value, and so on. So let's plot an EC, let's assign top genes. So if you see this just selects the top 6 genes, and we'll just plot their gene expression using Viling plots. There we go. So you can see that they're upregulated in CD16 compared to CD14 cells in general. We can also plot a feature plot, and we can see the gene expression mapped onto the UMAP. So you can see that the expression is much higher in CD16 versus CD14 cells. So the Wilcoxon test is good for quick exploratory analysis, and it has the advantage of being robust to outliers and non-normal distributions, as most gene expression levels in single-cell RNAseq datasets, and it's good for identifying marker genes with large effect sizes. However, you might have noticed these hyperinflated p-values. One of the main reasons for this is that we're treating each cell as independent when they're not. Yes, we filtered to only select control samples, but our control samples came from different lupus patients. And even if you just had one single sample that came from one patient or one mouse, and you wanted to compare 2 cell states, your cells would not be independent since they originated from the same patient or the same mouse or whichever tissue of origin. So cells from the same donor are not independent. They share genetic background, environmental exposures, and technical processing as well. So that's what we call pseudo-replication, and it can lead to overconfident p-values and non-reproducible results across studies. So yeah, Wilcoxon tests will give you an idea of cell state markers, but it's not the best way to do differential gene expression analysis in single-cell RNA-seq datasets. So what do we do then? As we mentioned in the previous video, there are other approaches that have been designed specifically for these cases in the context of single-cell RNA-seq data. Let's start with MAST. MAST, or Model-Based Analysis of Single-Cell Transcriptomics, is a statistical method designed specifically for analysing single-cell RNA-seq data and identifying differentially expressed genes between groups of cells. So why is MAST special? Well, single-cell RNA-seq data is very sparse, meaning many genes have zero expression in many cells. This makes traditional statistical methods less reliable, and MAST addresses this by using a 2-part hurdle model. So part 1 focuses on detection and is discrete, and models basically whether a gene is expressed in a cell, yes or no. And this accounts for the dropout effect, so when a gene is actually expressed but not detected. Part 2 of our model focuses on expression level, so continuous data, and models basically how much the gene is expressed, only obviously in the cells where it's detected. And these 2 parts are combined to give a more accurate and robust estimate of differential gene expression. Moreover, MAST allows us to adjust for technical covariates like sequencing depth, so total RNA count, percent of mitochondrial content, batch effects, or even the donor ID, which we said was a big source of variation. Okay, so let's get to it. We're going to use the integration of MAST in the Seurat package, which is very easy to implement, but if you have a more complex experimental design, or if you want to fit random effects, then I would recommend using MAST directly. You can find more information in their publication on GitHub. I'll leave the links below. Okay, so our first step is to create new identities combining cell type and condition. So we're combining the cell types and the condition, which is stimulated or control, and we'll just show you how this looks like. So these are all our unique values in that new column we created. So if we just run head to just check that, you can see that a new column has been created that combines both the cell type and the condition. Okay, and now we're ready to run MAST. It'll be really easy using scRNA. We'll use the find markers function again and test use set to MAST with the latent variables parameter, allowing us to include variables like ncount RNA to control for library size. We could also include other latent variables, so batch, mitochondrial content to improve the model. And again, if you want more control over the model structure, random effects, and so on, just run MAST directly. Great, so let's first select T cells. In this case, we're going to compare CD4 naive T cells from the control condition versus CD4 naive T cells that have been stimulated with interferon. So we've subset the scRNA object, and now we'll run find markers on our subsetted scRNA object, setting our comparison identities. So identity 1 should be our group of interest. That's why we set it to stimulated. So we're comparing stimulated versus control, with control and CD4 naive T cells being our base. So let's run this. It'll take a little bit. Great. So once it's done, you can of course save your results. You can save the table of differential expressed results, which in this case are stored in MAST_results. But let's have a look at our top MAST results. If you want to see more genes, you can print head or tail to see the lower part of our data frame. But in any case, you can see we still get hyperinflated p-values as we're treating cells from the same donor as independent. So let's just visualize our top differentially expressed genes. As you can see, quite a big difference between stimulated and control. And again, we can plot these on the feature plot. So here we go. So we've split our UMAP between control and stimulated conditions, and we have a single gene in each row. So this is IFIT3, and we see a high expression in stimulated cells versus control cells. And same with IFIT6. There we go. So as I mentioned, we still see hyperinflated p-values because we're treating cells from the same donor as independent. And in our previous method, we saw that pseudobulking is a great way to tackle this issue. So Pseudobulk approach aggregates cells by donor and cell type and condition, and then applies bulk RNA-seq methods like DSEQ2 or EdgeR, and it's currently considered best practice because it properly accounts for donor-to-donor variation. Okay, so let's get to coding. So first we'll attempt Pseudobulk using the scRNA aggregate expression function across stimulated and scRNA annotations, which contains our cell types. So this will just aggregate our counts. So in this case, we get one count per stimulated condition and scRNA annotation. So now we'll run DSEQ2. So we again use find markers and test.use DSEQ2, and we'll get an error. Group 1 has fewer than 3 cells. So this is because when we do Pseudobulk by condition only, we don't have any replicates. And without replicates, DSEQ2 cannot estimate dispersion. It's like trying to compare the height of normal and diabetic mice with a statistical test when we only have one value per group, so one normal and one diabetic sample. So in this case, since we've aggregated the expression by condition and cell type, we only have one replicate per condition. Essentially, each cell type condition combination, so for example, CD4 T cells stimulated with interferon, appears only once. So when there's no replicates, DSEQ2 cannot estimate dispersion. We need at least 3 replicates per cell type and condition. So what we're going to do is incorporate sample level metadata, which you can, we can download from the SCRA page, basically. So we'll just incorporate that metadata to our SCRA object. So let us see, we do a couple of steps to make this smoother. This is all taken from the SCRA webpage, so feel free to take these steps slowly and understand what it's doing in every step. But essentially, we've now added another column to our metadata, which has the donor ID. So which patient did it come from? And we'll remove cells without donor IDs, or donor ID equals to unknown, and then we will reset the cell identities to our cell types. But if we want to check how many patients we have, we can do this with table, donor ID. So we have 1, 2, 3, 4, 5, 6, 7, 8 different patients. Great. So we can re-aggregate our samples, this time using the donor ID as well. And now each group has replicates. So as you can see, we have incorporated the donor ID to our identity, essentially. Great. And now each group has replicates, and DSEQ2 can be applied through find markers. And we can use test.use DSEQ2, which will use DSEQ2 to do our differential gene expression analysis. And again, if you want more control over the statistical tests in the model, I would recommend you to use DSEQ2 independently, as with MAST. So build your own DSEQ dataset with more flexibility over design formulas and contrast. If you're familiar with bulk RNAseq data, then you should be able to do this fairly easily. But let me know if you'd like to see a video on DSEQ2 analysis. But let's visualize our DGE results. So again, we can print the top differentially expressed genes. We can also order our data frame by P adjusted value and print the top differentially expressed genes from DSEQ2. Again, we can use SCRA's volume plot, and as you can see a big difference in expression between the stimulated and control samples, at least for the top differentially expressed genes, as we expect. But again, you can plot your own box plots or volume plots for the top differentially expressed genes. And of course, you can use your results to do pathway enrichment analysis or gene set enrichment analysis. Before we wrap up, I'd just like to show you a very popular way of plotting our differentially expressed genes, which is with a volcano plot. You might already be familiar with this type of visualization. We're going to visualize our differential gene expression results through a volcano plot, and let's use, for example, our MAST results. So we basically show fold change in the x-axis and statistical significance in the y-axis. So let's first create a data frame with the columns we need. So first of all, we'll convert the p-adjusted value to log10 values. Then we'll mark significant genes as those that have a p-adjusted value lower than 0.05. and absolute average log2 fold change higher than 0.5. So this is only marking as true or false those genes that are upregulated and downregulated, and that up or down regulation is actually significant. So feel free to adjust the p-adjusted thresholds depending if you want to be more stringent or more lenient with your level of significance. With p-adjusted values, we usually would go a bit higher sometimes, again, depending on your results, but you could use a p-adjusted value of 0.1 even. If you use p-values, then I would say the most common thresholds are 0.05 or 0.01. Great, and then we're adding a new column called differentially expressed, which basically marks genes as up or down or non-significant, depending if they're upregulated or downregulated or not significantly up or downregulated. Great, so we'll just print the results so you see how this looks like, and we can also get a count on the number of genes in each group, up, down, and non-significantly expressed or differentially expressed. Great, so additionally we'll select the top genes for labeling. We'll order our genes by p-adjusted value, and then we'll select the top 20 genes that are significantly up or down regulated by the thresholds we set before. And those will be the genes that will be labelled in our plot. Again, you might want to highlight specific genes, or you might want to highlight only certain, downregulated or certain upregulated genes. So feel free to change this part of the code to adapt to whatever you want to show. Okay, so now we're ready for our volcano plot. We will use the MAST results data frame with our differential gene expression results. Then the x-axis has the average log to fold change, and the y-axis will use the log 10 p-adjusted values. And we'll color the results, or we'll color, our genes by the diff expressed column, which has up, down, or non-significant. And then we'll use the package ggRepel, so the function geom text repel, to label the genes we selected, which are stored in the top _ genes data frame. Great, and the rest is more for prettiness purposes, so just to make our plot a bit nicer and adjust the labels and the titles for the x and y axis. Great, so here you go, a very nice volcano plot. Again, you can adjust thresholds for significance, log to fold change, and even apply custom gene filters. Nice! So hopefully this tutorial gave you an overview of how to apply 3 differential expression methods in scRNA. Wilcoxon for simple comparisons, MAST for covariate adjusted single cell models, and DSEQ2 for pseudobulk analysis across donors. Each method has trade-offs, so Wilcoxon is fast and simple, MAST handles noise and sparsity well, but DSEQ2 gives you more rigorous results to handle pseudo-replication in single-cell RNAseq, which we already mentioned is a big problem. So that is all for today. Squid-tastic. As always, thanks for watching. Hope you found this useful, and do let me know if there was anything that was a bit unclear, or if there's any topics you'd like to see or to expand upon. Have a squid-tastic day, and see you in the next one.